thank you very much for coming. I'm very pleased to introduce the winner of this year's Mid-Career Award, Professor Neil Burgess. As we heard in the very interesting symposium this afternoon, Neil's work on phonological working memory, on spatial navigation, and on episodic memory has not only deepened our understanding of those particular phenomena, but it's also shown the way of integration between the methods and the purposes of traditional experimental psychology and computational modeling, systems neuroscience, and cognitive neuroscience. Particularly his work on grid cells and episodic memory has connected up work on animal neuroscience and human cognition. Neil's PhD, which was awarded in 1991, he did at Manchester in theoretical physics. But even at that time, he collaborated with Graham Hitch on work on phonological working memory. But aside from that period at Manchester and a one-year period afterwards in Rome with IBM, he's been remarkably loyal to UCL. He did his first degree at UCL and his first postdoc with John O'Keefe there. And then since that time, he's had an unbroken series of highly prestigious independent research fellowships from the Royal Society, from the MRC, and from the Wellcome Trust. And he's currently Wellcome Trust Principal Research Fellow, Professor of Cognitive and Computational Neuroscience, and Director of UCL's Institute of Cognitive Neuroscience. So Neil has got great ancestry, including Graham Hitch and John O'Keefe, but also he's someone who invests generously in the intellectual and personal development of the early career researchers who work with him. So he's had a very positive effect on the careers of many fine researchers, some of whom contributed to this afternoon's symposium. They include Rick Henson, Tom Hartley, Chris Bird, and Hugo Spears. And Neil has, of course, received numerous glittering prizes and awards over the years, but none more important, in my view, than his receipt of the EPS prize in the year 2000. Now, it happens that I wasn't able to attend his lecture at that point, but I was for many years a UCL girl myself, so I happen to know that he played for the UCL psychology football team, very creditably, and that he had a delightful nickname among the members of that football team. He was known as, well, we're going to have a conversation about Huxley, and I may be at liberty to disclose what his nickname was at the dinner this evening. But for the moment, it is with great pleasure that I ask you to join me in welcoming Neil Burgess to give the 16th Mid-Career Award Lecture.
Well, thank you, Celia, and the EPS for giving me this award. It makes me feel very humble, and thanks to Aidan for organising such a generous and frankly embarrassingly positive session just before. Yes, somehow in my mind it seems it calls itself the Midlife Crisis Award somehow. But yes, I'm in the middle of something. And I'm going to continue to tell you more about what many of the presenters earlier started to explain the background to. So hopefully that will help me make this comprehensible. And I really appreciate the fact that people in psychology take seriously this work, which is a bit interdisciplinary, certainly tries to explain human cognition and behavior, but also pays a lot of attention to what neurons are doing and computational models that try and link those things. And so I really appreciate this award as the one in 2000. And as Tom Hartley said before, actually, I really do think that, we know sort of superficially activity of neurons, neural firing, does generate behavior, very likely generates cognition, and I do think ultimately that that is the right level to try and understand these things. And so I'm going to talk about a couple of things in this lecture. So neural mechanisms, I take that seriously, like what are the neurons doing that make the thing, the cognition or the behavior happen? And I'm going to talk about spatial memory just because I think spatial behavior allows cross-species approaches, and also just acting in space, as we heard very beautifully from Kate Jeffrey's talk, space intrinsically has many constraints that you can use to your advantage to try and work out what representations are present. Okay, so on that theme, I'm going to talk briefly about frames of reference, and then some more of the stuff about place cells and bounded vector cells and grid cells, and their relationship, hopefully, to human spatial cognition and memory. So I quite like this experiment by Wang and Simons for its sort of simplicity in terms of trying to get at memory representations that are useful for memory. And hopefully many of you will be familiar with it, but I think it's worth explaining again. The participant sees an array of objects on a round table, and after a pause, they're going to be asked which object has been moved, so they have to remember their locations. But, well, and indeed, if nothing happens between presentation and test, that's this bar here, performance is very good. But this table can rotate by a certain amount, or people can be brought around to a different viewing position. And if the table on its own rotates, then people do relatively poorly. That's this bar here. Presumably, as these researchers thought, because the sort of visual snapshot that they'd encoded of the scene in front of them is now not matching the situation they present at test, because it's all been rotated, and that makes it harder. But the interesting conditions are these two in the middle, where I think they had assumed that if the table rotates by the same amount as the subject moves their viewing angle, then the visual scene is again the same at presentation and test, and they expected people to do well. But actually they do quite badly. They do a little bit better than if just the table rotates on its own. So even though they're seeing the same visual scene, they're not doing so well. And specifically, they're not doing as well as if they just move around to this new viewing point and the table hasn't moved. And in this case, they can perform quite well because they've automatically updated, if you like, where they expect all the objects to be, given that they know their own movement. And so this was strong evidence for the presence of something they called spatial updating, i.e. path integration, or updating how the world should look, given that you know how you've moved within it. And they also, there's also a bit of evidence for visual snapshots. Nobody would deny that we remember visual scenes. And so that's evidence for two kinds of internal representation. And one thing they said about this spatial updating was that people, they assumed, were representing sort of egocentric pointing vectors from their body to where these objects were in space. And these you can update automatically as you move around. But one aspect of this experiment I didn't like was the lab was visible behind the table. And so they could also be remembering these object locations relative to each other, which clearly they are doing, and that's not manipulated, but also relative to external things in the world, in the rest of the room. And they had only considered egocentric representations, either visual representations or representations relative to the body. And it's possible that allocentric representations, locations relative to the external world, were also playing a role here. So this is an experiment done by Hugo. Hugo Spears was my first PhD student at UCL, and what we did was to have the same manipulation where there's an array of objects on a table. The table can rotate, the person's viewpoint can change, but also this is done in darkness, and these are luminous objects, and the person wears a blindfold when they're not being tested or being shown objects. There was a cue card here which was also luminous and could also rotate the same amount. What we saw was basically that if you compare the presentation situation with the testing situation, it could be that the scenes are consistent, either in terms of any visual representation you stored at encoding, or in terms of where things should be, given that you know how you've moved and you know where they should be relative to you, or indeed they could be consistent with where they should be relative to this external cue card. And you can manipulate all three of these things independently in a 2x2x2 design. And what you find is that performance, if you do an ANOVA, shows simple main effects of consistency with all three of these representations, the visual snapshots and the spatial updating, but also consistency in terms of position relative to external cues. And so what this seemed to say, which was a relief to us because we were already, I have to admit, interested in allocentric representations and the hippocampus and place cells and all these things, is that in behavior you can see evidence of all three kinds of representations being present in the brain, and they're there and they can work in parallel and help you to solve these kinds of tasks when you have to perform them. And we saw no significant interaction, so it's almost as if they exist independently and can independently help your performance, although I don't think that's entirely true. That is how it played out in this experiment. Okay, so going on to these allocentric representations, and those of you who were here for the session earlier will be fully prepped on all of these, and those of you who've come in more recently, I'll go through a very brief introduction. Are found plentifully in the hippocampus, particularly in the rodent hippocampus, where place cells, as discovered by John O'Keefe here, are found. And now I have to work out how to... So those of you who didn't see the earlier pictures, this is a sort of cartoon representation of a place cell. A single neuron in the rat's hippocampus fires whenever it goes over into this place and doesn't fire whenever the animal's anywhere else. And it doesn't care which way the animal's facing. So these different place cells fire in different places, and together they're telling the rest of the brain where the animal is in its environment. Okay, so I was actually interested in memory following my PhD and working with Graham Hitch. And we know the hippocampus is involved in memory, for example, patient HM. And what I wanted to know was what the neurons were doing, and this seemed like a nice way of getting at least what's happening during spatial memory tasks. And we know from a lot of work, including Colin Lever here when he was in John's lab, that these place cell representations do have mnemonic properties. They show, for example, if you put the rat in two very different environments, they remap or show very different representations. So this cell fires in this environment and not this one. These two cells fire in this environment and this one, but not these two. And if they fire in both environments, they tend to fire in unrelated ways if these are very different environments that the rat clearly thinks of as different. That separation that develops very rapidly is seen still after, in the greatest example, 71 days later. So these neurons can learn to distinguish two environments and form a long-term representation that can be used by the rest of the brain. And by other people, this has been shown to depend, well, certainly the ability of place cells to show pattern completion in the sense of firing similarly to one set of cues or to another set of cues which have had some of the original cues removed. The place cells will still fire in the same way as long as there is NMDA receptors in these recurrent connections between place cells in CA3. And so we think there's a memory network there, and we can see it in the activity of place cells. They also show attractor dynamics which relate to this pattern completion, which I'll come back to. And so, as several people mentioned earlier on, if you take a single place cell and record from it in an environment that slowly that changes just shape and size, but it's still made of the same material, so you don't get any remapping, you can see that when a place cell decides to fire in a particular environment, it knows where to fire, at least in part because of the distances and directions from the boundaries of the environment around it, any extended objects. And if you pull these apart, then what is a small unitary field in a small environment can actually, you can pull apart what seems to be the influences of separate distances and directions from different walls into different firing things, which all coincide in the small box, but you can pull them apart at least temporarily. So this led to what Tom has already told you about, the boundary vector cell model, which is proposing that indeed there are detectors of distances and directions around the animal of extended environmental features like boundaries, or indeed any environmental feature. If it's there at the right distance and direction, then one of these proposed input cells will fire, and a collection of these input cells tuned to different directions and distances around the animal, and I mean allocentric directions, so north or south or east, doesn't depend which way the animal's facing. So enough of these cells come into the hippocampus then just by virtue of some place cells getting a random selection of some of these inputs, the conjunctions of firing of these inputs where they happen to coincide in space can drive the firing of a place cell in that place. So most simply, if a place cell tends to fire in the northeast corner of a square box or a circular box, it's probably getting some inputs from boundary vector cells that are detecting things to the north, and they would have firing rate maps like this. They fire whenever there's an environmental feature to the north of the animal, or one from the east. And then when they both fire, they drive the place cell to fire, and so it always fires in the northeast. And that's the simplest model that we could come up with for explaining this kind of data. And then, as Tom also mentioned, these proposed boundary vector cells were found by Colin Lever in the cerviculum, and very similar looking cells found by the Moses in medial entorhinal cortex, although they call them border cells for reasons which are not so clear. Anyhow, these various examples of these cells responding to environmental features. So each column is a cell. This shows a cell that fires whenever there's either a wall here or here that's south of the animal. So it could be an inserted barrier or the edge of the box where there's a wall, or even the drop at the end of a table, which is also a barrier. The rat can't jump off it, it's too high. And if you pull the two tables apart, there's an environmental feature here, the gap between the two tables, and it can actually jump across, but the cell still fires whenever that's to the south of the animal. So there's these boundary vector cells. Here's a particularly nice example from Steve Poulter and Colin Lever, which shows firing at a bit of a distance from the south wall, and then when a row of wine bottles are put into the box, it fires at a distance from that environmental feature that's been introduced. Okay, and even more recently, actually, object vector cells have been found by Jim Neerim in the hippocampus and by the Moses in entorhinal cortex. And these are cells which are very much like boundary vector cells, except they seem to be tuned to only respond to small things, whereas a boundary vector cell would respond to any environmental feature, big or small. Obviously, the big ones have a bigger impact on the firing of the cell because they fall into the receptive field more often. These seem to only respond to small objects. And here's 4 examples from Jim Neerim's lab and 3 examples from the Moses lab. And as with the boundary vector cells, they don't really care what the boundary is. They represent the existence of a boundary, whether it's a drop or a wall. These objects here, the little white circles, they could be different objects. These cells are not telling you the identity of the object. They're just telling you where it is as a vector, an allocentric vector from the animal. Whenever there's any object, small object, at that vector from the animal, then the cell will fire. And so you see these sorts of patterns. And so it seems like this sort of encoding things at an allocentric vector from your body is a major part of the inputs that come into the hippocampus and make place cells fire. Okay, so armed with all of this knowledge about what individual neurons can do in the brain in terms of representing where you are in space, and indeed where everything else is in space, because that's what it's relative to, we could try to make a neural level model of memory. So that's the idea. And we're starting with spatial memory because there's all this nice data telling us how individual neurons respond in space. So before we leap straight from place cells and boundary vector cells into human memory, we have to go back a moment and think about the egocentric representations. So I've concentrated on these allocentric representations of where the rat is, or indeed, as Kate mentioned, which way it's facing, the head direction cells, which depend on location or orientation relative to the environment. As we saw from the first experiment that I described, very important to cognition is egocentric representations, and indeed much of the brain, if we were recording in sensory cortex or motor cortex or parietal cortex, we'd be seeing lots of egocentric responses, cells that care about where something is relative to your eye or your body or your head. And in terms of behavior, this famous experiment by Biziak and Luzzati is nice on representational neglect in some patients who had grown up in the towns in Milan and were very familiar with the town square. And they had a right-sided neocortical damage, including parietal areas and prefrontal areas. So when they were asked to imagine this square that they knew very well, either standing at the cathedral facing away from it and to describe the scene that they could imagine from that orientation, then this particular patient described these buildings marked with red dots. And they all came from the right-hand side of that viewpoint, showing their neglect for the left-hand side of space, in this case for the left-hand side of imagined space. But if the same person was asked to imagine facing the other way and describe the square, they now described all these buildings on the right-hand side of this viewpoint, predominantly, showing that somewhere in their brain, probably the medial temporal lobe, they have a full representation of the layout of where all the buildings are on this square. And it's just when they're trying to call that into imagination to describe it that they're neglecting one half of that scene. And which half that is depends on which way they're facing in their imagined Milan town square, because that determines what falls on the left or what falls on the right. Okay, so equally, as I said, there's these various allocentric representations of location and orientation, which we'll come back to. There's also many more cells showing an egocentric response, and I'm just going to talk about the parietal lobe, where very interesting responses are seen that, classic visual responses, for example, a neuron will fire whenever there's a visual stimulus, a certain location away from where you're fixating. That's typical also of visual cortex. Or indeed, cells that code for your left-right turns as you make a certain route. There are lots of egocentric representations, and I'm going to come back to these because these parietal representations here, recorded by Richard Anderson in the 80s originally, showed an interesting feature, that they were visual cells, visually responsive cells that fired whenever, for example, this cell likes to have a visual stimulus at this distance and direction away from fixation. So the visual stimulus falls on a particular part of the retina, then this cell will fire, like a good visual responsive cell. But actually the firing rate at which it fired depended on the gaze angle of the animal. So if the animal was gazed to the left, even though the retinotopic location of the stimulus was still the same, the response would be higher than if it was gazing to the right and doing the same experiment. And so these gain field neurons that he calls were actually showing a conjunctive response to the visual input and its retinotopic organization, multiplied by something to do with gaze direction, so that you get a big response or a small response, depending on which way you're looking. And the same group later on, significantly later on, showed that you found the same kind of modulation also by the direction of the head, not just the direction of the eye. So the direction of the head in the world could multiply the visual response of these cells. And it turns out that this particular kind of conjunctive representation of a sensory sort of representation, visual in this case, multiplied by something to do with the angle of the head, is particularly useful if you're thinking about coordinate transform. And Alex Pouget, working with Terry Sanowski, pointed this out, because I'm not sure Richard Anderson had really fully appreciated that these sorts of networks where you have a bunch of neurons that respond to, say, the visual angle, and also multiplied by the angle of the head, they, together, that combined representation can translate for you either directions, egocentric directions like these visual stimuli. They can translate them. You can have a network of these neurons, and because you've got a full set of neurons that encode for visual angle and, in this case, head direction, you can then read out the allocentric direction from the animal from this kind of network. Or, if you know the allocentric direction, you can read through this network the other way and work out the egocentric direction. So what this is doing is just taking simply in populations of neurons and doing what's a simple transformation for you or I, that if I'm facing north and something is on my right, then it's going to be east. Or if there's something on my east and I'm facing north, I know that it's on my right, and vice versa. It's just doing that calculation for you. But it does it in a very natural way that you can encode in neurons. So if there's an input to this set of neurons from head direction, it could translate either way between egocentric or allocentric directional representations. And this is going to be very useful when we're thinking about human memory, if we think, as in the Biziak and Luzzati experiment, that people have an allocentric representation of where things are, say, in the Milan square, north, south, east, and west buildings relative to each other. And then we try and imagine, we try and generate some memory recollection of being there. We need to generate an egocentric scene that we can actually imagine. We need to know what's ahead of us and left and right of us. It's not good enough to know what's north and south and east and west. You can't visualize that. And so these gain field neurons will play a role in the model that I'm going to describe. And I should say, just to get ahead of myself, that this is a model that I started working on with Sue Becker in 2001, and we've slowly developed, and the final version that I'm going to show you is Andrzej Bikansky's work, who's currently in the group and in the audience somewhere. So if we want to think about representing the environment, how is an environment around us represented in memory? We could think about looking top-down on a whole population of neurons that are all tuned to respond to environmental features at different distances and directions. And so these are the receptive fields, these little blobs, of lots of different neurons that we're going to represent them as living, these neurons, in a circular sheet of neurons. In the brain, they could be organized wherever they like. But if we were to look down on a topographically organized sheet of neurons by their receptive fields, so these are their receptive fields, and this is, on the left here, is always going to be egocentric representation. So if the rat is facing this way, that's ahead. So on these neurons here respond to stimuli that are ahead of the animal, human or rodent. So if the agent, maybe we should call it, is here in an environment that has a sort of boundary of this shape, then that would be represented in this population of neurons by firing shown like this. So these neurons in this part of the sheet are the ones whose receptive fields are being driven by the presence of an environmental feature to fire, and that you can see is encoding the shape of the environment around the animal. And these are egocentric, up is ahead, but we could also represent the same thing in boundary vector cells, where what they care about is where things are north, south, east, or west of the animal. So here we would see a representation like this, because although the agent is oriented this way, what matters is where these boundaries are, north, west, south, and east. And we'd see firing like this on this sheet of neurons. So I'm hoping people understand, and what I'm trying to do is visualize what would happen if you could look down on the part of the brain that was representing the environment allocentrically, somewhere near the hippocampus perhaps, and egocentrically somewhere in the parietal cortex perhaps. And of course, to make our model complete, we're going to have to use these gain field neurons to try and translate between them. So if I know which way I'm facing, I can rotate these representations and work out where things are allocentrically from where they are egocentrically, or vice versa. So I could think about perception. Perception is always egocentric. It matters where the light comes in on my retina, for example, for visual perception. So I've got these bottom-up input, if you like, perceptual inputs here represented in that array of neurons, and I can put it through this transformation layer, which is a lot of gain field neurons. And if I know my head direction, I can work out where these environmental structures would be around me, north, south, east, and west. And by north, south, east, and west, I don't mean sort of actual compass direction. I mean north, south, east, and west as referenced by these head direction cells that signal the orientation of my head in the world, in a way which Kate Jeffrey explained very nicely earlier. So this would be a way of getting perceptual information about the environment into the medial temporal lobe, and perhaps stored in long-term memory. And there's a reason why it might be useful to store allocentric representations in long-term memory rather than egocentric representations. There's one advantage, which is that if it's, say, the Milan town square that you're very familiar with, you want to remember it and be able to recognize it, whether you come in from one direction or a different direction. You need to know the overall layout of it in a way that's not dependent on which way you happen to be viewing it at the moment. Okay, but equally, this transformation layer works in both directions. So we could also think about, if you like, top-down processing, where now I want to remember something from long-term memory. It's an allocentric representation. I can pass it through this transformation layer and then generate a visual scene of what the town square should look like if I'm facing in a particular direction. And again, that direction has to be encoded by head direction cells to tell me what's going to appear ahead or left or right in this visual scene. And so that's the basic outline of what we're going to use to try and make a model of memory. Okay, so we need some more details, unfortunately. This is the translation circuit. Here are the head direction cells. This is the egocentric representation. Here are the boundary vector cells and object vector cells, allocentric representations of the contents of the environment, the extended features and the individual objects. And here are the place cells. And so, as I said, there are connections between place cells in area CA3, so that they can do pattern completion. And also we assume that there's connections between these boundary vector cells and object vector cells and place cells, so that if you do identify where you are, I'm supposed to be outside the cathedral, then those connections can reactivate the boundary vector cells, which will tell you where are the other objects, where are the other large extended features around me in this square, and object vector cells that could do the same for any small objects. And finally, because boundary vector cells and object vector cells seem, somewhat surprisingly, just to encode the presence of an object or extended feature at a particular distance and direction, irrespective of what its identity is, we also have to have some identity neurons to associate to these neurons so that I can remember that that big thing over there is actually the chemist or whatever it happens to be. So we need some, what we think would be perirhinal neurons that encode the identities of these objects and boundaries. And now if we connect these all together, we can make this a long-term memory system, like a Hopfield model, if people are familiar with computational models, which will show pattern completion so that if I know where I am, I can reactivate the rest of the information about what is in the environment around me. And then using this translation, I can generate an actual scene and imagine being there. Okay, so that's how this is going to work. And in a familiar environment, because I've already learned these connections here by walking around the environment several times, all of this does is, you can think of it as ensuring that you generate a coherent scene. In a way, you can think that you've encoded lots of stuff in long-term memory, and there's lots of ways you could try and pull it out and imagine it. But actually, the spatial constraint is quite strong. To generate a coherent scene, you have to just pull out all the appropriate bits of information consistent with being in a single location, which is where the place cells are. And you have to generate a scene consistent with being in a particular viewing direction, which is determined by the head direction cells. And these are also, as Kate Jeffrey mentioned, thought to be arranged in an attractor structure, so that both place cells and head direction cells only have a single peak of activity representing a single location and a single direction at any one time. And that ensures that you generate a coherent spatial scene. Okay, so what I'm going to show you in a minute, sorry this is a bit of a busy slide, is one of Andrei's simulations of what happens in your brain when you go into a familiar environment and encounter a new object, or at least an object in a new location, and store that in your spatial memory, and then wander off and then try and remember where it was, what happens in your brain. I'm going to show you lots of top-down views of these arrays of topographically arranged neurons. So on the left, egocentric representations. On the right, allocentric representations. Here are place cells. This is the agent in a simple square environment approaching an object which it's not seen there before. This depicts the sort of sensory input. So whatever is ahead of the agent is thought to be visible to it and can come in and drive these egocentric perceptual representations, if we're doing perception and encoding, of objects, egocentric location relative to the agent, and of extended features. And also, because we've got head direction cells telling us which way we're facing, we can go through this translation circuit and generate a boundary vector cell or an object vector cell representation of the environment that's independent of our orientation. And we can learn associations between these and the place cells so that this becomes a long-term memory where we can do pattern completion and retrieve what was in that scene. Okay, these are the perirhinal neurons that indicate either the identity of the north, south, west, and east walls or the identity of this one object. So what I'm going to show you now is what happens as this agent wanders down here and then comes back again and notices this new object for the first time and tries to encode it in its medial temporal lobe representation of boundary vector cells, object vector cells, and place cells. So first of all, you should see the agent is moving, so the place cell representation is tracking that. And as it rotates, the head direction cell representation tracks that. And now it's approaching this object, which it can see. and here are the boundaries in front of it that it can see. Now I stopped the video here because it's encoding this object, and so we assumed this was a familiar environment, so the connections between the boundary vector cells and the place cells and the boundary identity neurons were all fully formed, consistent with this environment. So the right boundary vector cells for the dimensions of this environment and its shape and size had already had their connections learned to the appropriate place cells for the rodent or person being in each of the different locations. But there have been no objects encountered before, so now there's some object vector cells that are being driven by this perceptual input via the perceptual representation here, translated into an allocentric object vector cell representation. And we can now associate these neurons that are active with the place cells in that place and with the identity of that object, which we assume we get through the ventral visual stream to perirhinal cortex anyway. So now we've learned those connections, we can do another simulation where the agent continues on its way and at a certain point is reminded, where was that object? You need to recall from memory your encounter with it and imagine the scene. So now the agent continues to move and then turns away. It's going to do something else. You can see the place cells are tracking its location, head direction is tracked. It's going to get over here. This is the perceptual input. Now it's being prompted to recall the encounter with the object. And what's happened is that the identity neurons, perhaps it's the object's a set of keys, has been driven to fire at a maximum rate. And because of the learned connections, that makes the object vector cells fire and the place cells fire for the location where encoding happened. Those are the ones that are associated with these neurons here. And because the place cells here start firing, that also can reactivate the boundary vector cells to recreate the shape of the environment around you at the encounter when you were here. And then this activity can go through the translation circuit in the top-down manner and impose in some parietal region an egocentric representation of the scene ahead of you and left and right of you, and even actually behind you, because it's all generated from memory. It doesn't depend on perception now. And of course, the object vector cells get translated to their egocentric form, so that this scene you can also see or imagine where the object was, and then you can go back there and pick them up or whatever you want to do. I'm just going to show one more simulation, which just makes the point that actually, even though this simulation was explicit memory, try and imagine the scene where you were when you encountered the object. If you look closely, because of the recurrent connections that we've put in this medial temporal lobe bit, even when we're doing sort of perceptually driven encoding and exploring, you get the perceptual input drives some activity, say, in the object vector cells, and that drives the activity in the place cells. But the recurrent connections from the place cells also will try and reactivate, to a lower extent, these boundary vector cells consistent with the rest of the scene around the animal. And this might be an explanation for why in a familiar room, even though you can only see ahead of you, you're still aware of where the walls are behind you because you've made a memory representation of that environment. Okay. Does anybody have any questions about that simulation? I imagine, well, I didn't think anybody would say yes. Save your questions for the end. But I hope you've understood some flavor of at least what Andre was trying to achieve in terms of explaining behavior and cognition in terms of what would actually be happening with the neurons. Okay, and as James showed, we sort of know where these cells should be in the brain. These translation circuit, there's also cells, again, like cells in retrosplenial cortex as well as posterior parietal cortex. That could be there. We think the hippocampus, constrains the place from which your viewpoint is generated. This retrosplenial, but actually parieto-occipital sulcus region is doing this translation. And then maybe the egocentric representations might be in the medial parietal regions, which are associated with imagery and fMRI experiments. And so we can sort of build up a way to interpret what kinds of activity that we tend to see, say, in fMRI when people are remembering what's happened, we've got a way of interpreting what the neurons in these regions might be doing in terms of contributing to the function that's happening there. One point that I want to make is this idea of generating a coherent scene. Although it was the basis of this model with Sue Becker, it's become quite popular in terms of people talking about scene construction or episodic future thinking or planning. The ability to generate and imagine a coherent spatial scene is obviously very useful and not just restricted to remembering exactly what happened. If you can generate a scene which is consistent with a single viewpoint, a single direction, and make sure it's coherent, you can use that to imagine all sorts of things. And that could be a more general function for this network, which you often see both during episodic memory, but also during imagination and future planning. The other point I want to make about retrosplenial cortex is that it supports this egocentric allocentric translation, which obviously has its own uses, both during encoding and retrieval. But more than that, in the medial temporal lobe, we've got these head direction cells and place cells and perhaps grid cells, which are these internally generated allocentric representations, which have to be associated with sensory input from the environment. Otherwise, they would drift. They would never remain accurate. So you always have to link your internal representation, say, of where you are or which way you're heading, to environmental sensory input. And the environmental sensory input is always egocentric. So you always have to do this translation if you want to associate your internal representations with sensory evidence, if you like, from the world. And that has some consequences. For example, you'd expect stronger associations to form with stable sensory features so that they're always coinciding with the same head direction, the same location. And that might explain why, say, Eleanor Maguire's group tends to see more activity in retrosplenial cortex when you're navigating around when there's big permanent things there rather than small impermanent things. Okay, so if you put somebody in the scanner and ask them to remember what happened in a particular location, you tend to see these patterns of activity in these regions, and now we have some speculation for what those neurons might be doing. Tom Hartley already mentioned this experiment. Just the basic place cell and boundary vector cell model makes some predictions about where people will look for things in terms of just where will the place cell firing pattern most match that at encoding. But we've also got these egocentric representations, and as Tom said, in that experiment there was evidence that people are trying to match their point of view as well as responding at the right distances and directions from boundaries. In one particular example, if I remember it correctly, one person matched the view they had at encoding when they made their retrieval response, but where they put their response was matching the distance to the wall behind them that they couldn't see, having expanded the environment. And so you can see there's multiple systems at work here. As in that original experiment that I described at the beginning, there's multiple parallel systems here, egocentric ones, allocentric ones, and they talk to each other, but they're probably also independently trying to inform behavior. And how, for example, frontal areas arbitrate between which ones to pay most attention to and actually guide behavior is another interesting question. Okay, so this kind of model relates to other previous models, but is specifically sort of spatial in the nature of the representations involved, and that gives it a bit more detail in terms of knowing about those representations. But in essence, it's just like Ma's original model of the hippocampus and memory from 1971. There's recurrent connections in the hippocampus, which allows pattern completion, and this allows reactivation of neocortical representations of what happened. And that's all good. It's just we've gone into this in a bit more detail in terms of the actual how space is represented. And indeed, this pattern completion here in the hippocampus is a key aspect, and you'll see how our attempts, which I think somebody mentioned earlier on, with Aidan Horner, to look at standard episodic memory for remembering a person and a place and an object, how you can see pattern completion. I think James talked about this. Again, as you'd expect from this kind of model. It also gives one explanation for why, if you get hippocampal damage, like HM, then you become amnesic. But also, if you get damage anywhere along this Papes circuit, you typically have problems with episodic recollection. And this is one reason. There are many other reasons as well why that would happen, because as Kate pointed out, the head direction cells exist in this circuit from the mammillary bodies through the anterior thalamus and into the medial temporal lobe. And so severing that circuit means you're unable to impose, in this model, a viewing direction on your allocentric stored knowledge. And so you won't be able to generate a nice image to imagine. You might be able to retrieve data in other ways, but you won't be able to produce a nice viewpoint-dependent imagination if you lesion your head direction system, which exists along this circuit. Okay, so I want to cover a couple of things, and I'm aware that time is running short. We had several nice presentations about PTSD, post-traumatic stress disorder, and this dual representation theory. I'm going to try and say something briefly about that. To recapitulate what James said, there's an idea going back to Lynn Nadell, actually, and championed by Chris Bruin, who's another collaborator, that you could think of two different aspects of memory when you're thinking about what happens when a traumatic event happens. You might think about sort of explicit, declarative, contextual information that depends on the hippocampus and associations, and other areas like the amygdala and sensory cortex, which can actually store emotions and simple sensory representations that might be very vivid in the context of a traumatic event, like the knife or whatever it is. And this system here is normally associated to this system, so that normally you can deliberately recall the context and remember what happened. But in a very traumatic event, this system may have stored information very strongly in a way which can pop up, if you like, in the absence of weakened hippocampal contextual representations, and thereby be an intrusive thought which might give rise to post-traumatic stress disorder like symptoms. And so this dual representation account basically assumes that the trauma is upregulating these item-based and affective representations and downregulating these abstract or contextual representations. And it's this imbalance that leads to problems in the future. And we could compare that to a unitary model, which is just that intrusive traumatic memories are just very strong autobiographical memories. It's a unitary system, but something gets strongly encoded because it's so salient. And so James talked about this week, and here's our sort of context storing system, although items and identities are here in perirhinal cortex. And if we add to that the amygdala and sensory representations in the insula, then we can think of two competing representational systems that under trauma, it might be that this red system here dominates and this one is somewhat inhibited, and you get these strong affective and sensory representations that can pop up without contextual control. So James talked about some predictions of that idea. One more would be that, and Emily Holmes gave a lovely talk about how one could disrupt the consolidation of sensory representations after trauma and therefore produce fewer flashbacks by playing Tetris to make a competing representation or to disrupt the storage of the original representation. But equally, under a dual representation theory, we should be able to maybe boost the hippocampal encoding and favor strengthen the green system rather than the red system that I showed you, and that should also reduce the ability for these negative events to intrude on us. And Dewar showed quite nicely, and various other people since, that brief wakeful rest can facilitate, we think, the hippocampal consolidation of neutral memories. And so we thought we would try to see if giving people brief wakeful rest after seeing a series of these horrible videos that Emily talked about would change the amount of intrusive thoughts that people get subsequently when they keep a diary for the next week. So this is Lone Horlick and James did this experiment. So they see some horrible videos. They do either a wakeful rest manipulation or a working memory task. Then they keep a diary of intrusive thoughts. We also test their memory. And what was seen in this first experiment was that intrusions indeed were decreased by the wakeful rest just after encoding, whereas there was a slightly opposite effect, though not significant, on explicit memory performance when you're shown scenes from the video and have to recognize them. And we also replicated that with another task here, a provocation task, a blurry image from the film that might prompt you to have more intrusive thoughts. We find essentially the same thing as in the first experiment. We get reduced intrusions after wakeful rest and a slight but non-significant increase in memory performance. So Our manipulation does suggest a dissociation in that it has an effect on the intrusions, but not on the explicit memory, as with several of Emily Holmes's experiments, and therefore provides support for this dual representation account, which we're now making a little bit more mechanistic in terms of what might be happening in the brain. Okay, so I can't avoid saying something about grid cells. So I'm going to skip over. I hope most of you were at the session earlier, because what I'm going to skip over was covered there. We think that grid cells are probably driven by self-motion to some extent. This internally generated regular firing pattern, it's hard to imagine it is driven by environmental input, although there are some environmental influences, whereas place cells we think are driven by environmental inputs like boundary vector cells. But the point is the two kind of representations have to talk to each other, because we want, if you're purely relying on self-motion, your representation will drift relative to the environment. If you're purely relying on sensory input, then if you switch the lights off or you're not paying a lot of attention, then this representation will drift. It makes sense that they talk to each other, and grid cells and place cells do project to the relevant regions to do that. And so we thought we would test whether what most people believe is actually true by using virtual reality. So what I'm showing you here is a mouse virtual reality system that we've just developed. I think it's pretty much the only two-dimensional mouse virtual reality system that exists. The mouse is here, and it gets a little drop of milk through this tube whenever it crashes into a stripy pole in this virtual environment. These stripy poles appear randomly, but there's a place over here where the stripy pole no longer appears, but you still get reward if you go there, to show that it knows where to go to get reward, and it can remember where places are in this virtual environment. As it moves, it becomes much more obvious how the environment works. So the mouse is running on a styrofoam ball, and that drives the visual projection on lots of screens and on the floor. And what you can see is that this location here that had a cross is not marked, but the mouse knows to go there every fourth trial. Sometimes it goes there anyway, but every fourth trial it can't see any other stripy poles, so it goes to that location and gets a reward. While it's doing this task, which is the whole point, we can record, and I should say this is done with Francesca Cucci and John King and Guifeng Chen and Yi Lu. So while the mouse is doing that, we can record from all of these cells and indeed see that we can see normal place cell firing and grid cell firing and head direction cell firing, and they rotate appropriately when we rotate the virtual world, so that we know we've got control over the firing of these cells. So what we can do is we can change the gain between how much you have to run and how fast the visual projection moves to see whether these spatial representations are more influenced by how far I've run or by how much the visual world has moved around me. If we do that by changing the visual gain, either doubling it or turning it, dividing it into 2/3, what you can see is that for grid cells, these manipulations, basically, if you look in visual coordinates, they look a bit stretched one way, and if you look in motor coordinates, they look a bit stretched the other way. So if you calculate a sort of motor influence score, there's a range. Basically, grid cells are responding both to how much the mouse has run and by how much the world has moved, although there's a bias towards paying attention to the motor input. Whereas with place cells, the bias is more the other way. Actually, the visual coordinates of place cells look fairly good, whatever you've done to the gain, how far they have to run or not. And so what we see is that indeed it's true that grid cells are more influenced by self-motion cues and place cells more influenced by environmental cues, but they are probably talking to each other and both are influencing both representations. So I want to skip over that, and I'm just going to finish with a comment on if grid cells, we think that grid cells can actually talk to place cells and update the place cell location that's represented by self-motion inputs. So that's obviously useful during navigation, but also in terms of memory, we might think that that allows you to move the viewpoint location that you're going to generate an image from. So I might be able to imagine what the Milan town square looks like from just outside the cathedral, but I could then also imagine what will happen if I move in a certain direction or go around the corner, and that would be helpful for planning what to do, or maybe for retrieving some sequence of events that happened to me in the past. And so the grid cells would fit into the model like this, and as Alexa Constantinescu described very nicely with Christian Doehler and Caswell Barry when they were both in my lab. We managed to work out a way of a sort of proxy for detecting the presence of grid cell activity in the fMRI scanner in the entorhinal cortex here. But also, as Alexa pointed out, it appears in other regions which she related to value and other things. But this network, also the default mode network it's known as, is usually active during autobiographical memory. So it suggested that maybe movement of viewpoint in this system is important for episodic memory or autobiographical memory. And so Aidan, who arranged the very lovely symposium just before the break, he did an experiment where we did the same kind of task where you have to explore in a virtual environment, find some objects, remember where they are, and go and get them. But now, before you go and get each object, you spend a time just imagining going in that direction. And then you do actually go in that direction. And what we can see is here's the paths of a participant, and these are where the objects were. We can see the same six-fold modulation by running direction of activity in entorhinal cortex, both when you're actually going in different directions, but also when you're imagining going in those directions. So it is evidence that maybe these grid cells could be contributing to imagined movement of viewpoint. Okay, so finally, to link with Alexa's talk, as has been pointed out, the hippocampal cells, particularly in humans, represent all sorts of abstract concepts, often with a nice small tuning curve, just like a place cell. For example, the concept of Jennifer Aniston, whether you see her or see her name or whatever. And as David Tank's group has shown, that place cells, or things like place cells, will respond in a task where the rat hears a sequence of tones and has to respond at the right point to get a reward. You see neurons that respond for particular values of that signal. And finally, in Alexa's experiment, it seemed like there was this grid cell-like representation of movement in an arbitrary conceptual space of the lengths of the necks and legs of arbitrary birds. Amazing experiment. I really didn't think that would work. But together this probably shows that, having understood how these things fit together to represent where we are in space and perhaps to remember where things are in space, we might be looking at actually a much more general system where you have sort of state information. It could be any kind of concept, things that look like Jennifer Aniston or places like this, and also a way of representing movements in that kind of conceptual space, or the transition structure between these places. And this system would in principle be useful for representing conceptual knowledge of any kind that you needed to organize in a way that you could navigate through in a continuous vector way. So if I wanted to be able to search my memory space for all things that, on a gradient of how much the people like UCL, and on another gradient of how much money they have, then I could find the ideal donor. If you want to organize your, I don't think that very much, but I know the provost does. If you need to store your information, and we have all sorts of information stored in long-term memory in a way that's searchable, you probably want to be able to impose on it continuous organizational structures that you can navigate within for search, for finding the right elements in your memory. It may be that we're beginning to look at that sort of mechanism here in our spatial studies. So here's a conclusion. Maybe using all this stuff we know about space, mostly from rodents, we can create a neural level understanding of spatial memory and imagination, and maybe then even more generally conceptual knowledge. And I really must thank all of these marvelous people that I've worked with in my group and not actually in my group. And I haven't mentioned any work with Graham Hitch, although Celia did mention. I started off working in psychology with Graham, and he was the one that introduced me to it and sort of got me interested in memory in the first place. So thank you very much.
Thank you very much, Neil, for that splendid overview lecture. Since it's now 7 o'clock, I think we should go with the sometime occasional tradition of the Society and not follow a prize lecture with questions. But Neil, you'll be coming to the dinner tonight. Presumably you'll be outside immediately afterwards. So if anybody would like to discuss any of these issues with Neil, they will have an opportunity to do so. Sarah White, one of our local organisers, is going to very helpfully show us with a map how to get to the Case restaurant for dinner. But just before she does that, let's thank Neil once more for a great lecture.